Hi everyone, welcome and good morning. My name is Layla Abulhawa, and as most of you know, I am the primary contact at Integral Molecular for RVP sales and technical support. Before we start, I want to cover a few housekeeping items. All attendees will be muted during the webinar, so please use the Q&A box at the bottom of your screen to type in any questions you have. Don't hesitate to type in questions as they come up. If we don't get a chance to answer your question, we will follow up with an answer by email. The webinar will be recorded and a link to the recording will be emailed to you. Now, I'd like to go ahead and introduce our team members who will be presenting today. We have 4 members from our virology team. Tab Sullivan, who is a senior scientist, Haley Crawford, who is our virology project leader, Nolan Fox and Mallory Fouch, who are our research associate. We're also joined by Sharon Willis, one of our co-founders and the VP of Customer Relations. Also, Ben Durant, co-founder, president, CEO of Integral Molecular. Next, I'll hand it over to Ben Durant, who will give a background of our company and an overview of what we're going to cover today. Ben?
Great, thanks, Layla. And thanks for everyone for joining us. I know there's a lot going on in the world and in COVID in particular. We're trying to keep up with all of the changes that are happening in the fight against COVID, and I appreciate everyone joining us. We've been working on viruses and envelope virus-like particles and reporter viruses for the past 10 to 20 years, making our lipoparticles, which are envelope eVLPs, for 20 years, and reporter viruses for 10 years. We've published over 50 manuscripts on viruses. So when the SARS-CoV-2 outbreak happened, of course, last year, we were very engaged very quickly to try to fight and provide tools for everyone to fight against the virus. We've been working on SARS for the last year with everyone else. But before that, we've been working on dengue and Zika, Ebola, Marburg, HIV, Hep C, influenza, chikungunya, other viruses for the past 20 years. A lot of the company was founded based on virology, on virus technologies. So this is a continuation of what we do every day. Next slide, Layla. Yep, thanks. So today we're going to be talking about a few things. Of course, our RVPs, just a little bit of background in case you're not familiar with them. We'll go through that pretty quickly. We'll talk about cell lines, some of the quality control and protocol tips that continues to evolve, trying to make the use of the RVPs even better. And a lot of that has to do with how the cells behave. Some RVP tricks, particularly for aliquoting. People have asked about that, how you can store the RVPs even better. Of course, we'll talk a lot about emerging variants of RVPs. That's in the news and very much in the top of our minds, how to keep up with and even anticipate some of the RVPs, some of the viruses that are emerging in the world. We'll talk a little bit about how the coronavirus is being used on some of our other platforms for epitope mapping and specificity testing. And then we'll get to questions and answers, and we'll spend a lot of time on Q&A. We have some questions that have already come in and some frequently asked questions that we'll talk about first, and then we'll open it up to live Q&A to answer as many questions as we can.
Good morning. Thank you, Ben. I'm Haley Crawford. I'm the virology project leader, and today we're going to tell you all about our RVPs for everyone who's new. Our RVPs look exactly like the virus that we're studying. In terms of the COVID-2 RVPs, they express the spike protein on the surface. And then there's an option of one of 2 reporter genes, either Luciferase or GFP. These RVPs have a lentiviral core and a lipid bilayer. And we have optimized them for high expression of the spike protein on their surface. You can use these RVPs in antibody neutralization studies for screening of sera and they can be used in high throughput assays. We tend to work in 96 or 384 well plates. Another advantage of these RVPs is that they are safe in a BSL-2 environment. They are capable of one round of infection, and then they express the reporter gene, and that's it. And that's particularly advantageous for SARS-CoV-2 because it's an airborne virus. As I said, there's two different types of readouts, either Luciferase or GFP, and these RVPs go through a lot of quality control steps before we send them out to you. So two things we do with the RVPs to QC them, and you can do too, is infectivity and neutralization studies. So you can either use serum or antibodies in your neutralization studies. The figure on the left shows the readout for Luciferase RVPs. The unit is relative light units. For GFP, the unit is the percentage of infected cells, which is the percentage of cells expressing GFP, which gives you an indication of how many cells were infected. And then if you add your antibodies or your sera to the RVPs before you add them to their target cells, and the antibodies are neutralizing, they will prevent infection of the target cells. And then you'll get a nice neutralization curve like this. We have also developed an ACE2 stable cell line, and we ship these out with every free sample request and with some of our orders. And we have shown that over time, the ACE2 expression on the cell surface is maintained. The figure on the right shows that the amount of ACE2 on the cell surface at passage 2 is the same as at passage 30. And we have all of our cell maintenance protocols available online. Just quickly, a few tips and tricks based on questions we get from our customers. It is worth optimizing your cell density. We found that putting 20,000 cells into a well of a 96 well plate gave almost double the number of infected cells compared to 50,000 cells per well. So cell density is critical. In addition, proper passaging is critical to maintain the ACE2 expression. And again, all of these protocols are available on our website. We also get asked how many times the tubes of RVPs, they come in 1 ml aliquots, how many times they can be thawed and refrozen. As you can see, there is not an appreciable loss of infectivity when you use them first compared to if you've thawed them and frozen them again once. However, we do recommend that you only thaw them once, aliquot them into smaller amounts, and then flash freeze them and put them back at -80. And again, our protocol's online. If you follow it exactly, you shouldn't see an appreciable loss of titer. And then we also get asked how long you can leave your thawed RVPs out, either in the fridge or in the hood or in the incubator while you set up your assay. We have found that the half-life of thawed RVPs is about 4 hours. If you put these RVPs in the fridge, they will lose about 10% of their infectivity every hour. So we recommend working as fast as possible. Okay, onto the emerging COVID-2 variants. This amazing slide shows a phylogenetic tree evolving over time. And you can see that as this virus is evolving, it is picking up more and more mutations at a rate of approximately 23 substitutions per year. In the blue and purple on the bottom left, they're the 2 original Wuhan strains. The light blue and yellow in the middle are all the variants appearing over the world on the D614G background. And up in the top right corner, with the most accumulated mutations, we've got the South African, UK, and Brazilian strains. What's concerning about these variants is that they have multiple mutations on the spike protein, and this is leading to apparent increased infectivity. The effect of disease on disease severity of these mutants is not yet clear, but what we are noticing is that these variants have a selection advantage. They dominate locally, and then they are very quickly spread internationally. So the implication of mutations on the spike protein are twofold. One is that mutations can appear in the receptor binding domain and affect binding to the human ACE2 receptor. And the other is that they can occur on the spike protein where antibodies bind to them, possibly preventing neutralization. So some examples, the UK B117 variant's receptor binding domain has been reported to have a 10 times higher binding affinity for ACE2. And this virus is also approximately 60% more transmissible than the original virus. Models predict this UK strain will be the dominant one in the US by next month. The South African and Brazilian strains have this mutation E484K in the RBD, and this mutation has been shown to reduce neutralization by convalescent serum by more than 10-fold. And even more concerning is that cases of reinfection have been reported for all of these variants. And just last week, new reports from the UK have described this mutation, E484K, appearing in circulating UK and Wuhan strains. So we are keeping up with all of these developments. Last year, we developed the SARS-CoV-2 RVPs within a couple of months of getting the genomic data from China. We also then produced SARS-CoV-1 and MERS RVPs. They're all in the same family of coronaviruses. And then this year, we hit the ground running and have produced all these different strains that are appearing around the world. Our molecular biology team has amazing technology where we can just alter the spike mutation as soon as we get hold of a genomic sequence, and we can produce these RVPs. And then one of the critical things we do to test that these RVPs actually have spike on their surface and look like the real coronavirus is we do neutralization assays. So this is the D614G mutant. We add the CoV-2 monoclonal antibody that we have, and we can see that it does in fact neutralize these RVPs. If you take a look on our website, we are now updating our RVP offerings weekly. There is a long list of all of these emerging strains. A lot of them are available right now. They're available in GFP and Luciferase, and we have so many in the pipeline, and I estimate that's what our year will look like, just constantly adding to this list. In addition to these variant RVPs, we also have created our own library of alanine scan mutants. So these are RVPs that have an alanine at every single residue in the spike protein. I'll tell you a little bit more about that in a minute. And then we can also make any custom mutant you would like. You just tell us the sequence and we will create RVPs with those changes in the spike protein for you. So this is what we have available right now. The original Wuhan 1 RVPs, some with point mutations. We have the Danish mink RVPs and the UK and South African variants. We also have right now these 3 point mutations. One is that critical one I told you about earlier. The other 2 have been seen in Ohio and in Australia. And then in the pipeline, which will be ready at the end of this month or the middle of next month, we have the Brazilian variant, which was detected in Japan. We have the other Ohio strain, the Californian strain, a new South African variant, which has a deletion at 2 amino acids instead of a point mutation at 242. We also have a South African mutant, which has 3 critical point mutations in it. And then we have the new UK variant with the 484K mutation in it. And we will send this presentation to everyone who has attended this meeting, so you will all have this information. So what we do once we've produced the RVPs is we test that they are infectious. This is to determine that they are in fact expressing spike protein on their surface and that they can in fact infect cells to demonstrate how infected they are. So this is for Luciferase. Relative light units are on the y-axis, and here we have the UK, South African, and then the point mutants against our wild type control. And for every lot that we ship out, We also give you the Z prime value and the signal to background value, which are important to know for your assays. And then we also test the neutralization to see if these RVPs are recognized by the antibody, to see if the spike protein was expressed in a conformationally accurate way on the surface. And we've got some beautiful neutralization curves here. Similarly, we do infectivity assays with the GFP RVPs. And here again, you can see as measured by the percentage of cells expressing GFP, you can see that we titrated it. And when you buy a GFP RVP, we will send you the titer of that lot. And again, we have some beautiful neutralization curves for all of these variant RVPs. Then, as an added bonus, we have this Alanine scan library. So what we did is we mutated the entire spike protein at every single position and put an Alanine in there. And these Alanine scan mutants are available. We have pre-validated the expression of the spike protein on all of these RVPs. And you can see that on the figure on the left, the pale blue dots show that the spike was expressed on the surface of the RVPs. And then we took these RVPs, added them to target cells, and validated their expression. So this figure just shows the receptor binding domain, but we've done it for the entire spike protein. And this enables epitopes to be examined for neutralization escape hotspots. Something else we do at Integral is epitope mapping. We have a high throughput technique that reliably maps conformational epitopes at single amino acid resolution. So this is very similar to the Alanine scan library, where you send us your monoclonal antibody and we will tell you where it binds. We have thus far mapped 34 RBD mAbs, and we're also mapping mAbs that bind outside the RBD. There's a 4-week turnaround for these epitope maps, and we have had over 95% success rate. This figure on the left was published by our collaborators last month in Nature Communications. We mapped 2 mAbs for them, and we could determine which amino acids these mAbs bind to on the spike protein. And then the last platform to tell you about is our membrane proteome array. This is 6,000 human membrane proteins that are natively expressed in live or unfixed cells, and they are in 384 well plates. So what we do is take your antibody you provide to us, we measure the binding of that antibody to all of the cells in these plates, to the membranes on membrane proteins on the cells, and then with flow cytometry determine what they bound to. And this was recently used in this publication on the right, where this team did rapid selection of a human mAb by adding it to the membrane proteome array and detecting where it bound to the spike protein. Another crucial thing to do if you want to license a mAb for therapeutic reasons, if you provide us with your mAb, we will not only determine what it binds to, which would be your target, but we can also detect off-target binding, which is crucial to know if your mAb binds to something else in these 6,000 human membrane proteins. So that's all the data. Now I'm going to pass you over to our 2 research associates, Mallory Fouch and Nolan Fox, who are going to answer some of our frequently asked questions.
Hello, everybody. Thank you, Haley. My name is Mallory Fouch, and I'm going to be going over some general RVP FAQs. Currently, we have the South African, United Kingdom, and Danish mink variant available for purchase, as well as the N439K point mutation with and without the D614G background. At the end of February, we're going to have the Japanese, Brazilian, and Californian variant available. In March, the Ohio and 2 new South African variants, as well as the UK variant with the E484K point mutation. So can these RVPs be thawed, aliquoted, and refrozen? When working with the RVPs, we recommend working efficiently because once thawed, there is a slow decay of titer at 4 degrees. But when you get your RVPs, quickly aliquot them into smaller volumes and flash freeze them in an ethanol dry ice bath. For long-term storage, use a -80 freezer. For one aliquot, we do not recommend repeating the freeze-thaw cycle. And of course, our detailed freeze-thaw protocol is available on our website. Next slide.
Hi, I'm Nolan Fox. I'll speak on some frequently received questions in terms of cell use and our SARS RVP assays. What cells do we recommend using for SARS RVP assays? We will always recommend using our monoclonal stable cell line, which, as Haley mentioned, is the HEC 293T ACE2 expressing cells. These are available for purchase through our website and are also included in our free sample packages. We will also soon be offering a double stable cell line, which will express both ACE2 and TMPRSS2. Do you need any type of license in order to use our ACE2 stable cells? You do not need any type of license to use the cells that we provide. You can bank the cells for your own use, but do not distribute them. Can you expect any type of loss of ACE2 expression through subsequent passaging of our monoclonal cells? And at what passage should you expect to have to thaw a new vial of our monoclonal cells? It is very important to maintain our cells in puromycin in order to ensure ACE2 transgene expression throughout the entire population of cells. But when grown correctly under this puromycin selection, the receptor levels of ACE2 should not decrease with subsequent passaging. We personally carry our ACE2 cell line up to passage 25 without any appreciable loss in transduction efficiency.
What is the titer of the GFP RVPs? It's typically 1E6 transduction units per mil, but this titer will vary by the lot, and those values are reflected on the individual tech data sheets. What plates and seating conditions are best to use? This is dependent on the reporter of your RVP. For luciferase, we use a white poly-D-coated plate, and for GFP, we use a Corning black plate with a clear bottom. For our experiments, we add RVPs to the cells within 2 hours of seating the plate, and then detect our RVPs after 72 hours. For detection of the GFP RVPs, it requires either a microscope, high content imager, or flow cytometer. And to detect the luciferase RVPs, it requires a substrate and a luminometer. You should use reagents that are compatible with the Ranilla luciferase, and we recommend the ProMega Ranilla luciferase glow assay system. Do not use any Firefly luciferase reagents because you will not be able to detect our luciferase RVPs.
Is there a typical signal to background that you can expect to receive as an output when using our luciferase RVPs? This is dependent on the input you choose, but at a 10 microliter input, you can expect to receive a signal to background of at least 50 for any of the lots we provide. We also calculate Z prime statistics for each of the lots we produce, and these values can be found on the individual tech data sheets. Is there a specific antibody that we recommend using for neutralization assays with our SARS RVPs? We recommend using a VHH72 anti-spike antibody, which can be purchased through R&D systems. We've listed the catalog number on this slide. And in terms of our new RVP variant strains that we are producing, there is no change in the way you should handle them or the protocols. The standard protocols listed on our websites, which are used for the wild type RVPs, can be applied in the same way to any of the variant strains that we are producing. And now I'm going to hand it off to Layla.
Thank you, Nolan, and thank you all for joining us. I just want to thank all our customers for their continued support and trust in our virology team to produce the SARS-CoV-2 RVPs, as well as these emerging strains and variants. We are working very hard and we are very committed to providing these strains to you as fast as possible without compromising the quality that you've become accustomed to. So thank you and for trusting in us, and also thank you for your patience as we get quotes and information over to you. We will always keep you updated with the latest information as we have it, and we'll always be transparent with the mutations and deletions that we have in our strains.
All right, so now we're going to move on to the Q&A portion of our webinar. So if you have a question, please feel free to put it in the Q&A and we'll be sure to answer it. We have received some questions so far. So the first question that we have is for Tab. Can the RVPs be used to test cross-neutralization titers and convalescent serum from patients infected with different SARS-CoV-2 variants?
Thank you for the question. So the answer to that is that yes, the RVPs can be used for convalescent sera from patients infected really with any variant of the SARS-CoV-2 virus. And they can be tested for cross-neutralization across all of the variants that we have available currently, which were listed earlier in the presentation. And we have the table online where you can see all the currently available and upcoming variants that are available.
Great, thank you. Okay, next question is from Mallory. What target cells have these RVPs been tested on? I know you mentioned we talk about our stable ACE2 cell line, but what else have we worked with?
Yeah, so we have tested our stable cell lines with ACE2 and ACE2 and TMPRSS2 transiently transfected. We've also tested Vero or Vero E6 cells and CACO2 cells as well, but those don't show very good infection, or we haven't had very good infection with our RVPs with those cell lines. So we really recommend our stable cell line to provide the most reliable rate of infectivity.
Great, thank you, Mal. Haley, next question for you. You showed some lovely infectivity and neutralization data. And it looks like, you know, there is a difference in titer for the different strains compared to the wild type. So how would someone know how much to use in an assay?
So for our GFP lots, we will give you the titer, and it tends to work out at about 10 microliters of RVPs you add per well in a 96 well plate. For our Luciferase, we will give you signal to background and Z prime, and that will help you determine how much to add. If you're adding 10 microliters to each well of a 96 well plate, 1 mil will get you a whole plate.
Great. Okay, let's see. Next question is for Tab. What is the RVP origin? I guess which virus genetic backbone is used for making the RVPs?
Yeah, so we use a lentiviral backbone based on the HIV system. So that's what the viruses are based on. And I think you can find most of this information in the tech data sheet and MSDS of the virus.
Okay, here's a question that just came in, and Mal, can you answer this one? Is there any difference in accuracy or sensitivity for the GFP versus the luciferase reporters?
Hi, so I don't think there is any difference in accuracy or sensitivity of the GFP or luciferase. They're both fairly sensitive and accurate.
Yeah, and I think depending, a lot of that depends on your reader also, on how quantitative your reader is in the assay readout.
I can say, as well, I see another question coming in about how to read it. So just kind of following that up, we use flow cytometry typically for detecting GFP. Of course, you can use a microscope or a high content imager. I wouldn't necessarily recommend a plate reader, which kind of averages the fluorescence in a well. With cell-based fluorescent assays, that tends to be pretty low sensitivity. But again, a microscope, high content imager, or flow cytometer works really well. Going back to the other question, of course, GFP and luciferase are just very different readouts. So kind of, which one you use is dependent on which system makes sense for your screens. Luciferase can be very, very fast and very, very sensitive. GFP, you can see individual cells and count individual cells and use flow cytometers. And of course, with GFP, there's no extra reagent needed, whereas luciferase, you need the vanilla substrate. So different readouts for different purposes, but you're welcome to try both and settle on one, whatever works for you.
Okay, thank you for all the questions coming in. Here's another one. This one's for Tab. So the RVP neutralization assay seems to be more sensitive than the live virus print assay. The person asking this has seen this in many sample sets. Why do you think this might be?
Yeah, thanks for the question. It could have to do with the total viral input, or, I think that the sensitivity comes down to the number of spike proteins and the amount of antibody present in the sample. So I think that it can be calibrated to be more in line if you adjust inputs possibly to your print assay, but it probably comes down to the number of viral particles in the actual input.
Okay, great. Thank you,
One
Ted.
thing related to that too that we brought up last time, we didn't present it this time, but the correlation between RVP NT50s and live virus PRNT NT50s, is very good. A customer of ours published that a few months ago. We can make that available again if people are interested, but the correlation between the neutralization values in our assay, we're using RVPs versus live virus print assays, was very, very strong. I think the correlation was 0.9 or something close to that. So that's something that we watch carefully to make sure that the RVPs are reflecting the actual virus. Of course, we make them to be as close as possible to the actual virus. But the sensitivity, partly comes down to the reporter, and partly, of course, it's a very different assay. And as Tab said, the amount of cells you use, the amount of virus you use can make a big difference in the actual sensitivity of detecting neutralizing antibodies in serum. Okay,
great. All right, here's a question for Haley. Do the RVPs have the M and E proteins of SARS-CoV-2 as well as spike?
No, these RVPs don't have M and E. We just express spike on the surface.
Okay, great. Let's see. Next. All right, here's another one for Haley. This is with regard to safety. So how do you prevent these strains from infecting humans?
If you look at our MSDS sheets on our website, you will find all the instructions for safety and handling of these reporter viruses. They are very safe. To work within a BSL-2 lab, if you follow all the recommendations, you should be fine. And as I said earlier, they're only capable of 1 round of infection. So if you're careful, you should be fine, and they cannot establish infection in a human, and they cannot cause disease.
Okay, great. All right, so let's see, another question. Okay, for Tab, how are your stable cell lines optimized? How was that process done for optimization?
Thank you. So the stable cell lines we developed are optimized for infectivity using our RVPs. So, one of the things we noticed is that ACE2 expression, the higher the ACE2 expression doesn't necessarily correlate with higher infectivity. So we optimize on infectivity as opposed to ACE2 expression. Beyond that, as we've mentioned before, as long as you carry them following our SOPs very carefully, we have seen out to 30 passages, no appreciable loss in ACE2 expression or infectivity. So these cells should be stable for quite some time.
Great, thank you. Okay, another question for Haley. With all these variants coming out, what's the estimated timeline for generating variant and custom RVPs?
So we have got our timeline down to between 6 and 8 weeks now. Once we receive the point mutations you would like or the strain you would like, we hand this sequence to our molecular biology team. They create the plasmids. That process can take around a month, and then they QC them, give them to us. We produce the RVPs, and then we QC them. So up to 2 months until they're ready to ship.
Okay, great.
I will add too, we understand for everyone how important it is to get a hold of these variants as quickly as possible. So we continue to streamline the production process, but also the molecular biology process to prepare those clones. We're literally monitoring variants coming out on a daily basis, and when there's significant new variants that come out, we get those into our pipeline immediately. So we're trying to respond as quickly as possible. It does take some amount of time to do the molecular biology and the production and the QC to get it out to you, but we're trying to do that very, very quickly without losing any of the quality. We're even starting to, one of the things behind the scenes is that we're actually anticipating variants that could come out. We never know, of course, exactly what variants are going to emerge, but with the types of mutations that are coming out, we can make some predictions about what might be next. And we're creating some of those variants in case they do emerge. And if other ones emerge, we'll create those, but we may already have them in-house even before they become popular variants. So again, we're trying to speed things up even beyond the normal to get every one of you the variants you need for your tests.
And to follow up on what Ben just said, and something I didn't say in the presentation earlier, is that with all these variants coming out, we will soon be establishing a panel of variants where you will be able to buy approximately 10 mutants, 1 ml tube of each of them, so you can have a whole array to test in your assays.
Great, thank you. All right, here's another question that came in. This is for Tab. Can you comment on the correlate that there might be with utilizing a competition type assay with ACE and RBD spike plasma samples in an ELISA type based format?
Yeah, so that's a great question. And I think that what the two assays do is similar, but not exactly the same. So in an ELISA-based assay, you're really just looking at blocking binding between the ACE2 and the spike protein. And that can read out on some types of neutralization. But there are other types of neutralization that happen. We know that there are antibodies that can neutralize the virus that don't bind to the RBD. So it's probably not a direct blocking of that interaction. So the RVPs may give a more robust readout of the various types of neutralizing antibodies that can be found in sera and that can be used in therapies.
All right, another question that just came in for Haley. Should we acquire the GFP signal by flow cytometry or use a plate reader with appropriate filters?
We recommend using flow cytometry. As Ben mentioned earlier, if you use a plate reader, it detects the luminescence per well, and it's so much more accurate to use flow cytometry and detect the number of cells that are expressing GFP. It gives you a much more specific readout.
Okay, great.
But if you have a plate reader and not a flow cytometer, then you can use the Luciferase RVPs.
Okay, thank you. Question for Ben. Has anyone used the RVPs in experiments with animals?
I don't know the answer to that question. I've not seen any data from our customers or collaborators doing that, but I know we've talked about that in the past, but I haven't seen any data yet. But it certainly is a very feasible experiment to track where the virus is going in vivo.
Okay, thank you. Another question for Tab. What's the best way to avoid non-specific inhibition of RVPs by sera that lack the COVID-2 antibodies?
Yeah, that's a great question, and there's a couple ways. You know, some of it is preventing non-specific inhibition, and others is controlling for it. So the best way to prevent non-specific inhibition in serum is to heat and activate the complement in the serum. So, you know, we work with, I believe, a 65-degree heat and activation shortly before we use the serum in neutralization assays. And the other way to address this is to control for it. So we also have RVPs with a different viral envelope protein, so VSV, available that can be used to detect non-specific inhibition of the RVP system with a different viral envelope protein.
Okay, great, thank you. Another question for you, Tab. Can you comment on the double stable cell line ACE2 TMPRSS cell line that's going to be available soon? Are there any advantages to working with that?
So far as we've seen with our RVPs, there's no real advantage as far as kinetics of infectivity or the level of infectivity received that you'll see with this double stable cell line. I think the uses of the double stable cell line will be if you're interested in and you have specific questions about the role of TMPRSS2 in your system. So as far as using them to get a more robust infectivity, we're not seeing that in our hands.
Okay, great, thank you. Haley, I have a question on the morphology of our ACE2 stable cell line. And also, again, if you could talk a little bit about maintaining it, but is the morphology of those cells any different from the parental cell line?
The morphology of the ACE2 293 T cells is very similar to the wild type cells. The SOPs that we put up on our website will tell you how we have, how we found the best way to maintain them and maintain the transducibility of these cells. So yeah, they should look the same. And if you follow our protocols, they should work the same.
Okay, great.
And I'll add on too, you know, whenever you're doing virus-based assays, RVPs or otherwise, there's always two components, of course. You know, there's the virus and there's the cells. You can't really do a virus infection assay without the cells. So, you know, we can quality control the RVPs that we send out, and we can send you the cells to start, but the health of those cells growing in your lab is extremely important, you know, for the actual final assay. If your cells are happy, you'll get good results. And if they're the right density and they're growing and, you know, expressing ACE2, you'll have great data. If they're not happy, if the CO2 is off or the incubator is running dry or the media is old or they're too confluent, you know, lots of things can go wrong with cells. That definitely affects the assays. You know, we've certainly seen internally, we do, you know, we've run infection assays and something happens and the cells don't look good and the exact same RVPs don't give the right results or give poor results, less infectivity than we expect. But then when we replace the cells and or correct the problem, the RVPs perform beautifully. So, you know, if you're seeing any kind of oddness with your cells, whether it's the incubator, the cells, contamination, of course, the media, that's going to affect the experiment. So you want to make sure you're keeping your cells happy for all the RVP assays.
Great, thank you, Ben. Okay, another question. This one's for Nolan. Can you just reiterate the protocols that are best to use with the variant strains?
Sure, yeah. There is no difference in the way you should handle or the protocols you should follow when using these variant strains. The standard SOPs listed on our websites that you would have been using for the wild type RVPs are the same ones you should apply for any of the variant strains you receive from us.
Great, thank you. Okay. All right, here's another question for Haley. What is the genetic makeup of the RVPs? Does it contain more than just the RBD sequence? Is the full spike there? Any other CoV-2 proteins?
So the RVPs have the full length spike protein on the surface, not just the RBD. It's the full length spike protein, and the extracellular region is not truncated. So the whole thing's there. There are no other SARS-CoV-2 proteins. The nucleocapsid is lentiviral. And so, yeah, the only CoV-2 protein in these RVPs is the full length spike.
Okay, great, thank you. And then Haley, can you just again reiterate the two South African strains that we either have or that are coming along?
So the initial South African strain we produced had, which starts with 20C, as in it came out in 2020 and it's clade C. It has 9 mutations in it. We then discovered that there was another variant, which is called 20H. And it has the same 9 mutations, but instead of one of those mutations is actually a deletion of 2 amino acids. So we have that too, that is coming. And then on top of that, we are producing a South African mutant that is not seen in the wild, but it has the 3 most crucial South African mutations in it. Those 3 mutations are in both the 20C and the 20H. So we make that as well.
Okay, great. All right. That is about all of the questions that we have. I'm going to turn it over to Ben to wrap up with some closing comments.
Yeah, I'll just reiterate one other technical thing that was mentioned in the slides. You know, continue to use the Ranilla Luciferase reagent. The glow reagent is what we recommend. If you use Firefly Luciferase reagent, it won't work with Ranilla Luciferase. And if you use the flash type reagents, your signal goes down very quickly. The glow reagents last for half an hour to an hour with really consistent signal across a 96 or 3D football plate. So, again, we have that in our protocols for what we recommend. If you use that, you'll get more consistent results across a plate. And we have seen, people asking about that. So I just want to reiterate that. But again, thank you to everyone for joining to the webinar and for your trust in us, as Layla said, and providing you these reagents. We know we're part of a global fight against this virus, and we're happy and proud to be able to contribute to this. We also know that these variants are emerging very fast, hopefully not faster than we're all vaccinated, but we're trying to provide everyone these tools to test your antibodies, test your vaccines, test serum samples, so that you know what's coming, what's protective, and what to test against. As I said, these variants are coming so fast that we're monitoring it on a day-to-day basis, trying to anticipate what's coming next. There's only so much of that we can actually do, but to the extent we can predict where the virus is going and how it's emerging, we're trying to get these variants created as quickly as possible. Sometimes they're created in our lab even before they emerge, if we have the right bioinformatics kind of insight into the direction the viruses are going, the mutations that are recombining. Other times we have to wait to see what actually emerges and what becomes relevant for testing against, and that takes a bit of time to do, but trying to get that down to as minimum a time as possible to provide you those reagents for testing. So thank you again for joining us. We're here to support you. If there's reagents, services, products, or even scientific questions that we can help with, we're happy to talk about these things. At heart, we're scientists as well as you. We're working on papers, we're working on R&D projects, and we're providing products and services to you to enable that as well. So anything we can do to help, whether it's part of our commercial services or part of our scientific expertise, we're happy to talk with you and help move the world forward a little bit. Thank you.
Yeah, and please, any questions or comments, you all have Leila's contact information. Please reach out to her. She'll facilitate answering questions, setting up phone calls, and getting you all the pricing that you need and information to help your research. Have a great day, everyone. Thank you.
Thank you.